Good day. This is the Professor, CBMD PhD, and today's Tuesday, May 23rd, 2023. It is 9:22 and 2 seconds, a.m. Eastern Daylight Time, and today's research, bacterial barriers. So let's jump to it here. The breaking through bacterial barriers and chronic treatment-resistant wounds. Date May 22nd, 2023. Source: University of North Carolina. Healthcare. Using a new strategy, researchers were able to reduce the challenging MRSA infection in the wounds of diabetic mice by 94%. They were able to completely sterilize the wounds in several of the mice, and the rest had significantly reduced bacterial burden. Has to defend itself from our immune system and other threats, as aureus can band together, creating a slick, slimy forefield or biofilm around itself. The biofilm barrier is so thick that neither immune cells nor antibiotics can penetrate through and neutralize the harmful bacteria. Researchers have developed a new method that combines palmitoleic acid, gentamicin, and non-invasive ultrasound to help improve drug delivery in chronic wounds that have been infected with S. aureus. When bacteria are not completely clear from chronic wounds, it puts the patient at high risk for the infection reoccurring or developing a secondary infection. And this therapeutic strategy has the potential to improve outcomes and reduce relapse of chronic wound infections in patients. They are excited about the potential of translating this to the clinic, and that is what they are exploring right now. Researchers were curious to know if non-invasive cavitation, enhanced ultrasound, can create enough agitation to form open spaces in the biofilm to facilitate drug delivery. Liquid droplets, which can be activated by ultrasound, called phase change contrast agent, are applied topically to the wound. An ultrasound transducer is focused on the wound and turned on, causing the liquid inside the droplets to expand and turn into microscopic, gas-filled microbubbles when they move rapidly. The oscillation of these microbubbles agitates the biofilm, both mechanically disrupting it, as well as increasing fluid flow. Ultimately, the combination of the biofilm disruption and the increased permeation of the drugs through the biofilm allowed the drugs to come in and kill the bacterial biofilm with very high efficiency. Microbubbles and phase change contrast agents act as local amplifiers of ultrasound energy, allowing us to precisely target wounds and areas of the body to achieve therapeutic outcomes not possible with standard ultrasound. Researchers hope to be able to use similar technologies to locally deliver chemotherapeutics to stubborn tumors or drive new genetic material into damaged cells as well. When the bacterial cells are trapped inside the biofilm, they are left with little access to nutrients and oxygen to conserve their resources and energy. They transition into a dormant or sleepy state. The bacteria, which are known as persistent cells, in this state are extremely resistant to antibiotics. Researchers chose gentamicin, a topical antibiotic, typically ineffective against S. aureus due to widespread antibiotic resistance and poor activity against persistent cells. The researchers also introduced an antibiotic adjuvant, palmitoleic acid, to their models. As palmitoleic acid, an unsaturated fatty acid, is a natural product of the human body that has strong antibacterial properties. The fatty acid embeds itself into the membrane of bacterial cells, and the authors discovered that it facilitates the antibiotic's successful entry into S. aureus cells and is able to kill persistent cells and reverse antibiotic resistance. Overall, the team is enthusiastic about the new topical, non-invasive approach because it may give scientists and doctors more tools to combat antibiotic resistance and to lessen the serious adverse effects of taking oral antibiotics. As systemic antibiotics, such as oral or IV, work very well, but there is often a large risk associated with them, such as toxicity, wiping out gut microflora, and C. difficile infection. Using the system, we are able to make topical drugs work, and they can be applied to the sites of infection at very high concentrations without the risk associated with systemic delivery. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.